Okay, so it's 9 o'clock. Get started. Hello everyone to our today's Zoom seminar. It's absolutely my pleasure to welcome our guest today from all the way from Australia, Dr. Daniel Gray. Daniel currently holds a position as a full professor, also the head of immunology division in the Walter Elsa Hall Institution for Medical Research from Australia. A very, very brief introduction about Daniel. Daniel completed his PhD with Richard Boyd, studying how the stroma cells of the thymus governing the T cell differentiation. And then after which he continued to pursue his postdoc training in Harvard Medical School with Diane and Christoph, focused on how autoimmune diseases are initiated in the air deficient animal model. From 2009, Daniel was recruited back to Australia to the Walter and Elsa Hall Institute for Medical Research, and then work with Andrews Stresser on regulatory T cells and then how autoimmune responses are shaped by apoptosis, which I think we're going to hear a lot today. Daniel become independent PI in 2013 and then was appointed as associate professor in 2017. From 2023, just last year, he became full professor in the institution. Daniel's lab actually particularly interested how cell death process shape adaptive immunity, seeking to understand the molecular control of immunological tolerance responses to cancer and lymphocytic malignancies. He actually has really a big reputation in immune tolerance and then in also cell death and cancer immunology. His work define how immune tolerance checkpoints work at the molecular, cellular, and organismal levels with focus on how the genetic lesions give rise to disease-causing immune dysregulation. His lab's main contribution actually cover several different areas, which including how life and death decisions control regulatory T cell homeostasis, and then how the new molecular determinants of T cell differentiation and deletion. And third is how the thymic stroma imposes immune tolerance. Last but not least, his lab also studying how, the immunological machinery of anti-cancer responses. His lab has really did a lot of groundbreaking discovery to illustrate the machinery of immunological tolerance to improve human health, and then to make fundamental discovery about immune function, which create new diagnostic and treatment for disease. I happened to heard Daniel's talk a couple months before. It was super impressive, which I decided right away to invite him to on our platform. We very much appreciate your joining us today, and then thank you so much. We are looking forward to your talk.
Great. Thanks very much. It's a very kind introduction. I appreciate that. And good morning, good afternoon, good evening, everybody, wherever you are. And thanks for joining this seminar. And thanks for Chuan Wu for organizing this excellent series. I think it's a wonderful contribution to the community, making discoveries in immunology accessible to so many people. And so I really appreciate this opportunity to share some recent work from the lab with you. I'm located, as Chuan said, in the Walter and Eliza Hall Institute in Melbourne, Australia, or WEHI as we call it, which has a rich history in immunology stretching right back to Macfarlane Burnett's formulation of the clonal selection theory I hope you all learnt about in lectures. So Gustav Nosel's demonstration that one B cell will make one antibody, so an important test of that theory. And Jacques Miller's discoveries, of course, around the thymus and the distinction of T and B cells. And this focus on immunology continues with over 20 labs throughout this multidisciplinary institute still devoted to understanding adaptive and innate immunity. So to start off, I must apologize for this vague title, Aging and Drugs and Cell Death. And the reason is that I want to share two stories from the team, and they both touch on these three themes. And so the first is about the curious case of thymic involution and a new stromal cell feature
Daniel,
that we, oh, yeah,
Daniel,
can you
sorry.
see the,
I think we don't see your screen. Maybe you share it again.
oh, okay. Sorry. Let me try again. I'll stop the share and start in just a moment. Mhm.
Yeah, looks
How's
great.
that?
Yeah, looks great.
Okay. Apologies for that. But yeah, so the first story, aging, is about this feature, thymic involution, and this new feature we discovered. And if you get bored by this story, don't worry. Just wait a little bit and I'll share some data from another program, one that's focused on how new therapies target cell survival proteins and activate a rapid feedback loop in human leukemia that reduces the effectiveness of these targeted therapies. So to the first story, you can see that okay? Yeah. Good. As I'm sure you're all aware, the thymus is the first organ in the body that undergoes age-related atrophy, referred to as thymic involution. No one quite knows why this happens, but the consequences are clear. You can see in these sections here of human thymus that the lymphoid tissue in adults is replaced with fat, and there's a structural breakdown that culminates in the reduced production of T cells. You can see here from Daniel Dueck's TREC analysis, or T cell receptor excision circle analysis throughout life. You note the log scale. You have this rapid, also degenerative decrease in the production of new T cells. And this thymic involution and diminished T cell output begins shortly after birth and accelerates through adolescence and worsens from there. This is a phenomenon that's been observed in virtually all creatures that have a thymus. So this reduced production of T cells has been linked with poor adaptive immunity in the aged, particularly when confronted with new pathogens such as seasonal flu or SARS-CoV-2, for instance. And this is because the TCR repertoire narrows as we acquire more memory T cells at the expense of newly produced ones from the thymus. Without those new T cells coming from the thymus, the capacity to respond to new pathogen wanes. This has prompted the question of why thymic involution should happen at all. And there's been several interesting hypotheses put forward, but no one's really been able to come up with a way to test these. Some of these include that maybe it's just a way to conserve energy because it's a wasteful process to create and destroy so many thymocytes in this process. Maybe it's a way of reducing the risk of malignancy, so a T cell acute lymphoblastic leukemia, or immunity, as we know that the thymus has to screen a lot of potentially autoreactive cells. Maybe involution de-risks that. Maybe it's just dispensable. It's something that happens when it provides no evolutionary selective pressure to stop it from happening. Actually, Jacques ventured this theory, and it was one of my favorite for a long time, just something that happens. But the discovery I'm about to share makes us question whether maybe it's a more active process. So there's a few others, but regardless of the reason, a severe and clear impact of thymic involution can be seen in the setting of myeloablation, followed by hematopoietic stem progenitor cell transplantation for the treatment of certain blood cancers. This intensive therapy effectively reboots the entire immune system and is a critical parameter of success is how quickly patients can restore immunity. Unfortunately, T cell reconstitution is severely compromised in adults because of thymic involution, and that makes them highly sensitive to infection and the substantial morbidity and mortalities associated with that. Kids tend to fare much better because they still have an active thymus, and that their survival correlates with the ability to restore T cells. So what's needed is a way to reverse thymic involution in these groups or restore function somehow to reform that T cell pool. So in this first story, the question that motivated it was, what are the changes that are occurring during thymic involution that might drive this process? And we had a particular focus on the epithelial cells of the thymus, since they mediate all the key steps of T cell differentiation, and they're known to play a central role in thymic involution. We also know that the thymus is a regenerative organ. However, the capacity to regenerate reduces with age also. So following acute insults such as infection, stress, irradiation, the thymus of young animals can readily regenerate to restore normal function, or involution continues, but you get back thymic function. Represented in the gray lines, though, is the observation that in aged animals where the thymus is involuted, the regenerative process is incomplete. Prior studies had quantified that this loss of thymic tissue and structural changes are perhaps linked to this, but in general, these focused only on certain aspects of or regions and lacked a systematic way of tracking the thymic epithelial cells in situ. In this image I'm sharing here now sort of typifies why it's been problematic to study in the past. So in cyan, we've got a genetic reporter showing the cytoplasm of all thymic epithelial cells in the mouse thymus, and in magenta are the thymocytes. And you can appreciate that TEC form this complex three-dimensional network, kind of like a sponge, if you like, through which the thymocytes sort of percolate. And that maximizes the contact between the two cell populations and ensures that you get this exchange of information. However, thymic epithelial cells are actually relatively rare. They represent less than 1% of thymic cellularity. They're highly heterogeneous. You might have read recent papers from Diane Mathis and Jacob Abramson and others that demonstrated that there's a remarkable heterogeneity of thymic epithelial cells. And these isolation protocols that we use to study them by flow cytometry or single-cell RNA-seq, we know underrepresent the real diversity and number of thymic epithelial cells. So they've been difficult to study traditionally. So the way we try to get around this problem and bring to bear new imaging approaches that have emerged is to create a new TEC reporter mouse where FOXN1, a key transcription factor for thymic epithelial cells, drives a Cree that switches a dual reporter allele from a ubiquitous TD tomato signal shown here in the top left, which has a nuclear localization signal, so it's constrained just to the nucleus of cells expressing it. But the FOXN1 Cree causes a switch to occur whereby GFP, now shown here in cyan, is expressed again only in the nucleus of thymic epithelial cells. So this allowed us to quantify epithelial cells in a really nice way in situ. And so by confocal analysis, you can see here we could resolve nicely individual TEC nuclei, and we were able to confirm this with other markers. And this high density you see in the middle parts here represents the thymic medulla, the inner part, where these key stages of air-mediated negative selection occur, for instance. And then you've got this outer cortical region here where there's sort of sparser thymic epithelial cells. That's where the early stages of T-cell commitment and positive selection and so forth, that's where they occur. And then there's this outer subcapsule region where you get this lining epithelium. So having confirmed through this and a bunch of other studies that the reporter was working as it should, Kalin was able then to prepare and optically clear entire thymic lobes from these mice and subject them to light sheet imaging. And so this produced some spectacular results. One here is shown from the thymus of a young mouse. And again, in cyan, you're looking at the GFP signal representing thymic epithelial cells. Magenta is everything else. And if we create a mask to show where this higher density of thymic epithelial cells is occurring, I'll play that again if I can. You can appreciate that the thymic medulla is actually a relatively complex structure and highly interconnected. So in blue, you'll see in a moment, represents the thymic medullary regions that are connected to another one. And in yellow are those rare thymic islets, the medullary regions that are actually not attached to anything else, which is interesting because by section, you tend to predict that maybe they were isolated islets, but that's not actually the case. The picture is very different when we look at the thymus of an aged animal. So you can see here, first of all, the size is smaller because of thymic involution. But we were struck by these new features. You can see here, there's really high intensity band-like structures that kind of surround the thymic medulla in this involuted thymus. This is a feature we just don't see in the aged thymus. In addition, the thymic medulla had changed. You can see we've lost that interconnection, and you've got this recession into these small isolated medullary regions represented in magenta on the right here. You'll see in a moment. We highlight, yeah, that's the thymic medulla of the involuted thymus. You can see it's surrounded by this band-like structures of really high intensity GFP signal. So we wanted to know more about why that's the case. And so here we're just looking at confocal analysis of thick sections. Again, on the left is the young 2-month-old thymus. You can see beautiful, clear cortex and medullary regions. However, the involuted thymus is very different. Although we can still resolve cortex and medulla, you can see this high density GFP signal that's just not observed in the adult thymus. And this represents a collapse of thymic epithelial cells. Remember that beautiful three-dimensional network I was showing you earlier on? That's completely lost within these structures, such that now you can see in this higher power, the nuclei of these thymic epithelial cells are right next to each other, indicating that they're packed full. And it's hard to discern here, but we found that the thymocytes couldn't access these new microenvironments that are created by this high density of thymic epithelial cells. Now, I'm a skeptical type, and so I'd spent many years looking at the structure of involuted and young thymus, and I'd never seen anything like this. So when the PhD leading these studies, Kaylin Sao, when she first showed me these features, I immediately thought, okay, this is some artifact. Perhaps the reporter allele's been activated in mesenchymal cells or some other problem. But fortunately, Kaylin's very stubborn, or should I say rigorous. And so she went and tried to convince me that this is really a new feature, a new thymic epithelial cell type. And so here you can see that she's taken thymi from these involuted thymus from the age reporter mice and shown demarcated one of these high density tech regions here, and shows very nicely that the stains with a pan-keratin antibody. Keratin is an intermediate filament and is a key characteristic of all epithelial cells. She also went on to show they do not label with markers of fibroblasts or endothelial cells, other stromal cells in the thymus. And so they were clearly establishing them as an epithelial cell type in the thymus. Now, around this stage, some colleagues posted a preprint on bioRxiv that reported a single-cell RNA-seq survey of the thymic stroma from young and aged mice. Since they included cells from 18-month-old mice, we predicted this data set would include data from those cells that we were seeing in these high-density tech regions. And this analysis was led by a talented bioinformatician, Anastasia Kusa. She was working in Marcel Vandenbrink's lab and Jared Dudakov's group. And these data are represented here on this UMAP, showing thymic epithelial cells from either a 2-month-old mouse, shown in blue, or an 18-month-old mouse, shown in this red color. And you can appreciate that there are substantial changes captured in this type of analysis, such that we see the emergence of a transcriptionally distinct population, or at least two populations, in the thymic epithelial cells from the involuted thymus. That we just, this on the right here, we've highlighted these two populations, and we just don't see blue cells in there. Now, by comparing these profiles to previously published data, they could quickly identify all the known major epithelial cell types in the thymus, except for these two populations on the right-hand side here. So again, they labeled these age-associated TEX1 and age-associated TEX2, or AA-TEX. So these populations had a unique transcriptional signal, one that they couldn't find in any cell atlases throughout the body. And so we, consistent with their distinct location on these UMAPs, a survey of the expression of genes that are characteristic of the different subsets demonstrated that these AA-TEX on the right-hand side here had lost expression of many key genes. So for instance, AIR, for instance, is not expressed, nor key markers of cortical epithelial cells, or the recently described mimetic cell populations that are found in the thymic medulla. These age-associated TEX had none of that. And curiously, a key, like a canonical epithelial cell feature, the expression of EpCAM, a cell adhesion molecule, was lost in at least one of these populations, AA-TEX, indicating or perhaps suggesting one of the reasons why the field had overlooked this feature for so long. So these were very interesting findings, but we really needed to establish whether one or both of these AA-TEX populations might correspond to the cells that we'd found through the imaging. So one of the approaches we took was to use spatial transcriptomics to try and localize the AA-TEX. And so we're looking at sections of the 2-month-old thymus on the top here, or a thymus from an 18-month-old on the bottom. And using a Visium platform, we could identify, using a variety of markers, the outer cortex, shown in green, or the inner medulla, shown in magenta. If we then take the transcriptional signature of AA-TEX 1s or 2s and overlay them, you can see that, first off, we don't find these features in the young thymus, as we might predict. But in the older thymus, we see there's a few of these scattered regions with a much warmer color on this heat map, indicating higher expression of this transcriptional signature. Again, it's associated with the thymic medulla. Remember those high-density TEX structures we showed before seem to be next to the thymic medulla. And indeed, they overlap with one another, suggesting that both AA-TEX 1s and 2s are existing in the same microenvironments within the thymus. In another approach, we undertook a careful analysis of differentially expressed genes that might distinguish AA-TEX from their counterparts and tested many of these candidate markers. Many of them work, but the most definitive of these was claudin-3. Claudin-3 is a component of tight junctions found in the gut and other epithelial tissues. It's known to be expressed by some medullary thymic epithelial cell precursors, and that's the signal you can see down here. There's a few of these red dots indicating high expression in some cells in the thymic medulla. But by far the highest signal we observed was in these AATEC1, and insofar as nearly all of the AATEC1 cells had high expression of claudin 3. So if we take thymic sections now from these reporter mice, aged reporter mice, Kaylin's showing two examples here. She stained for claudin 3 in yellow, and you can appreciate that it overlaps beautifully with these high density TEC regions highlighted here again by their high density of GFP expression. And we see that virtually all the cells within these regions are expressing claudin 3. And while there's a few of these sparse medullary epithelial cells expressing claudin 3, by far the biggest signal we see in the involuted thymus is restricted to these high density TEC regions, providing further evidence that the AA-TECs that were identified in the single cell RNA-seq study correspond to the high density TEC regions that we resolve by imaging. So armed with this information, we could learn a little bit more about the phenotype of these cells using flow cytometry. So we took the thymus from young FOXN1-CRE, NT, and G mice, the reporter mice, and we looked at the expression of GFP in the stromal fraction. And consistent with all previous studies, all the thymic epithelium, so this is cells with a history of FOXN1 expression, were EpCAM positive. And if we gate on those cells, we can use markers LY51 to distinguish the cortical epithelial cells, or CTECs shown here, and a lectin called UEA1 that helps identify the medullary thymic epithelial cells shown here. In general, there's more MTECs recovered in these digests than CTECs. There was a different picture in the involuted thymus from these mice, though. The majority of the GFP positive cells were EpCAM positive. If we gate on those, you can appreciate that we've now got a population of LY51 and UEA1 negative or double negative cells. This is an unusual feature in TECs. And if we gate on this AATEC1 and plot claudin 3 expression, I'm not showing you here, but we found that that's where the majority of claudin 3 expression resided, suggesting that these indeed are the AATEC1s that we observed in our imaging and transcriptomic studies. But another feature that emerged was that there was an unusual GFP positive EpCAM negative population. If we gate on those and look for AATEC2 markers, in this case I'm showing you podoplanin, we find that they have high expression of this. So that corresponds to that transcriptional signature of the AATEC2 population I introduced earlier. Loss of this canonical TEC gene, EpCAM, and gain now of some mesenchymal traits like podoplanin. Okay, so now we've got some nice markers that will enable other investigators to look at AATECs. We wanted to understand whether this is a feature of the human thymus. And so we're able to query already published data sets from a 30-year-old donor and overlay the AATEC1 and AATEC2 transcriptional signatures on epithelial cell data sets from humans. And you can appreciate that we can recover a substantial fraction of cells from this involuted thymus that are characteristic of AATEC1 and AATEC2. And if we do a histological survey now, and this is from a thymus from a 50-year-old female donor, we stain that with TEC markers such as keratins shown here, or thymocytes, CD1A shown on the bottom here. You can see we can find these structures that are keratin positive, but exclude thymocytes, which is exactly what we see with these high-density TEC regions, indicating to us that AATEC are also a feature of the human involuting thymus. Okay, so one of the features you'll note, though, is that these AATEC regions exclude developing thymocytes, suggesting that they no longer support the key function of thymic epithelial cells. To learn why this might be the case, Anastasia analyzed the expression of transcripts that are associated with the function of cortical epithelial cells. So this is regulators of T-cell commitment, proliferation, positive selection, migration, and so on. And MTEC function as well. So I showed you they don't express air, but what about key differentiation cues such as rank receptor, lymphotoxin beta receptor, and others? Well, we find that in typically the AATEC populations do not express these genes, suggesting that they're unable to support the key steps in T-cell differentiation. If we do a broader gene set analysis, enrichment analysis, confirms that we have defects in key antigen processing machinery here. So this is an enrichment showing that while other thymic epithelial cell subsets have high expression of antigen processing machinery and MHC and so on, The AA-TECs tend not to have these same pathways active. Conversely, though, we see upregulation, particularly in this AA-TEC2 population, of an epithelial to mesenchymal transition state. It's a partial one, but you can see here on this barcode plot that many of the genes associated with an EMT signature are enriched in AA-TEC2, but not in other thymic epithelial cells. We were able to confirm that you have the loss of EpCAM, also E-cadherin is downregulated. And on the other hand, we have transcription now of mesenchymal genes such as vimentin. However, this transition doesn't seem complete because, as I showed you, they retain keratin expression and other key features of epithelial cells. But we think that this signifies something about the factors driving AA-TEC formation in the involuted thymus. However, something else we noticed that was, although AA-TEC didn't express genes required for thymocyte differentiation, they do have appreciable transcript for key TEC growth factors, such as FGF receptors shown up here, and also BMP receptors and the EGF receptor, suggesting they could still bind key growth factors that drive thymic epithelial cell proliferation and differentiation, factors we know are really important for thymic regeneration. So this prompted the hypothesis that perhaps AA-TEC impacts somehow on thymic regeneration. Maybe they're part of this reason why the involuted thymus can't regenerate so well. The way we modeled this was to challenge the mice with sublethal irradiation, which causes acute thymic atrophy, followed by a regeneration phase, an endogenous regeneration phase. And we can follow this first off by assaying the number of cells in the thymus, and we're plotting that here. And you can appreciate that in these blue data points, following a steep decline, the thymus of young mice regenerates relatively quickly, such that by 4 weeks, it's nearly returned to baseline. In contrast, the regeneration of thymi from aged mice, shown in red here, is slower. It's still not returned to normal levels, even by 6 weeks later. So there's a substantial lag in the regeneration of the involuted thymus compared to the young. So what we did then was try to look for AA-TECs using our reporter mice here and imaging approaches at day 0 compared to day 28, when we have the biggest difference in thymic regeneration between the young and old mice. And so here's some examples here of this analysis. So on the left-hand side, we're showing the 18-month-old thymus, and the AA-TEC are highlighted in cyan. And you can appreciate that, again, we've got this high-density TEC, mostly on the inner thymus, that are kind of scattered around the thymic medulla. It's not shown here. However, if we look on the right-hand side, this is the thymus from one of these reporter mice taken at 28 days post-irradiation. So it's beginning to regenerate, but it's not yet matched the baseline size like I showed you in those graphs. And the AA-TEC surface, again shown in cyan, you can appreciate looks like it's greater in volume. And so if we quantify that here, we're comparing the thymus from an untreated mouse to one that's 28 days after irradiation, shown in gray. The thymic lobe is still half the size, and both the medulla and cortical regions are reduced. But by contrast, the AA-TEC, the volume that's composed by the AA-TEC has increased by about 2 or 3-fold. This represents a change in the overall number of thymic epithelial cells. So MTEC, we're able to count those using our light sheet data and the confocal analysis and get a complete representation of TECs. We see the MTEC are lower, CTEC curiously is slightly higher perhaps, but by far the biggest increase we see during this impaired regenerative phase is an expansion of these non-functional AA-TEC cells. Okay, so we wanted to know why that might be, and we remembered the expression of these receptors for key growth factors. And so what you're looking at here is a summary of a cell chat analysis that Anastasia undertook. This predicts major signaling inputs and outputs among cells in the RNA-seq dataset, based on the single-cell transcriptomic data that takes into account receptor ligand pairs, but also the activation of downstream genes in cells that might be receiving key inputs. So if we take data sets from the thymic stroma taken at day 7 post-irradiation, so this is the early phase of thymic regeneration, Anastasia focused on pathways known to be promoting TEC regeneration. So we're looking at FGF signaling here. And the way to look at these plots is that arrayed on the outer part are all the various thymic stromal populations, all these different colors. And the AA-TEC 1 and 2 are shown here in cyan, and this mustard color shown in number 2. And we're comparing the data from 2-month-old mice with 18-month-old mice. Consistent with prior literature, we know that FGF signals such as keratinocyte growth factor are derived from the mesenchymal populations early, and they drive the proliferation of MTEX to restore thymic regeneration. So you can see many of these arrows going to these other TEC populations. The picture changes in the involuted thymus. So with the accumulation of AATEC1 and 2, you can see now many of these arrows are driving towards AATEC1 and 2, co-opting signals that would normally be going to other conventional TEC subsets. It was a very similar picture with BMP signals. Jared Dudakov's labs showed that these are a key driver of thymic regeneration. They come from fibroblastic and endothelial cell populations in the thymus, seem to sense the damage to the thymus and drive BMP signaling to thymic epithelial cell populations. But again, the emergence of AATEC seems to draw some of these trophic signals away from the conventional TEC subsets and perhaps depriving them of those key trophic factors. So these data suggest that AATEC co-opt these reparative cues required to support thymic regeneration. So to summarize this part of the study, we've identified 2 novel TEC states that emerge with age and have features of EMT while maintaining epithelial identity somehow. AATECs create an aberrant thymic microenvironment that does not support thymocyte differentiation, and these age-associated TECs seem to perturb normal thymic stromal interactions and impair regeneration following damage. If you'd like more, we recently published this study. There's a lot more data in these papers, but these are the key messages. And some of the implications are that, well, first of all, it identifies a previously overlooked aspect of thymic involution that may shed light on the processes that underlie the loss of function in the thymus. And if we come back to those hypotheses around why thymic involution happens, it's tempting to speculate that if involution is an active process to conserve energy or prevent autoimmunity, then perhaps these age-associated TECs are a key mediator of that process, something that forces the atrophy of the thymus for a good reason. So to put it another way, maybe the major function of this cell population is to cause thymic involution. Whatever it might be, though, we're curious now to understand the molecular details and the origin of AATEC so that we can ablate them or reverse these cell states to perhaps open new ways to regenerate the thymus and restore immunity in those immunosuppressed populations I mentioned earlier on. Okay, so now we're going to switch gears a little bit and discuss another study, one that's about the rewiring of cell survival processes in leukemia when they're under pressure from new targeted therapies. So for this study, it's important to review the pathways to apoptosis in cell. And there's 2 major pathways, the intrinsic or mitochondrial pathway, or the extrinsic and death receptor pathway. We're going to focus on the intrinsic pathway here, and a key step in this whole pathway that is almost used to be called the point of no return, but that's being revised as we speak, but is the permeabilization of the mitochondria in cells. When this occurs, the release of apoptogenic factors provokes the activation caspase activation cascade that then leads to dismantling of the cell in an immunologically silent manner. So this is a key step in the whole pathway. So it's subject to very strict regulation by, and it's predominantly from the family of proteins, the BCL2 family of proteins. Now there's 3 major factions in this family, and the key mediators are BAX and BAC. They can reside at the mitochondrial membrane, and their activation causes these pores to be formed, and that causes mitochondrial permeabilization. In healthy cells, their activation is restrained by pro-survival members of the BCL2 family, including BCL2 itself, a classic survival protein. But when cells are under stress or exposed to cytotoxic drugs, another group called the BH3 only proteins are activated and upregulated, and their job is to relieve this inhibition either by interacting with the pro-survival proteins to remove their inhibition of BAC and BACs, or by directly activating BAC and BACs through their activities. Now, while this pathway is hardwired into all cells, a complication is that there are many different BH3 only proteins and many different pro-survival proteins, and each have distinct expression profiles in cells and different response kinetics to various stimuli. So that variation is represented here with a bunch of different immune cell lineages, for example, shown and the various pro-survival proteins that are predominantly sustain their homeostasis that are highlighted. This complexity enables the differential regulation of mammalian cell types, but it's surprisingly hard to predict how different cells will respond to a particular cytotoxic stimulus. And this is important because resistance to cell death has long been recognized as a major hallmark of cancer, one that frustrates the efficacy of many therapies. Okay, so this is particularly apparent in hematological malignancies, as this table summarizes various B-cell cancers and the pro-survival proteins in this pathway that they upregulate and that they rely upon. Now, a game changer for the therapy in these conditions has been the development of small molecule inhibitors that can selectively inhibit the pro-survival proteins, BCL2, MCL1, BCLXL, and can cause apoptosis in these cancer cells. Now, the first in class of these drugs is venetoclax, an inhibitor of BCL2 itself. It has a remarkable efficacy in the treatment of chronic lymphocytic leukemia, or CLL, which is a relatively indolent, slow accumulating blood cancer that's typified by very high expression of BCL2. And that's why it was selected for the first clinical trials of venetoclax. And some of the data from that trial is shown here. This is a pioneering study from colleagues here at WeHi showing that there's steep decline in the leukemic burden in CLL patients that receive various doses of venetoclax. And in fact, in this study, it was established that one of the issues with using venetoclax is tumor lysis syndrome. It's such an effective drug that it needs to be delivered in a dose escalation fashion so that you don't induce tumor lysis syndrome, which can be life-threatening in the patient. However, there are ongoing challenges, and one of those is the emergence of therapeutic resistance. So the CLL can become resistant to venetoclax. This plot here sort of exemplifies the problem. What you're looking at patients who had a very good response to venetoclax, shown in red, so a complete response indicating that CLL cells could not be detected in the blood of these patients after receiving venetoclax. And they go on to survive for many years. On the other hand, patients that had a partial response, whereby surviving CLL cells were able to be found, they've somehow been able to resist venetoclax treatment, those tend to go on to have a relapse within just a few years. So in our study, we wanted to understand these early phase of venetoclax treatment and what it's doing to the CLL that might inform better treatment strategies to ensure that more patients have a complete response and are able to not resist. So for this, we wanted to capture the complexity of these apoptotic pathways in the context of all the major cell lineages that are in the peripheral blood of these patients. And so what we did was take a series of samples from over 20 CLL patients just before they went on to the next highest dose of venetoclax through their treatment. And we subjected these then to cytop analysis. So this is just a way we can do high parameter analysis using metal isotopes to detect antibodies rather than fluorochromes that might traditionally use for flow cytometry. This enabled us to unlock a whole suite of different epitopes we could detect to find various blood cell lineages, key immunomodulators, but also a variety of regulators of cell death and cell cycle within to visualize what the impact of venetoclax treatment might be on CLL and immune cells in these patients. And so this is just a quick schematic of cytop, which I won't go into. But what we could do then is take the data from all of these patients at all of the different time points and do FlowSum to cluster the various key populations. And here what I'm showing is a UMAP dimensionality reduction here. And I'm just highlighting the key cell lineages that we could find by overlaying the expression of CD19 and CD5 in this case to highlight B cells, and importantly, the CLL cells shown in various shades of blue here. We could also identify the T cell clusters shown down here, CD4s and CD8s, and also key innate immune players like myeloid cells and NK cells. But in our study, we wanted to focus on the CLL cells, and you can see here that you can identify them very clearly in a patient with CLL. We detect very few of these cells in a healthy donor. And the thing to appreciate is that at all doses of venetoclax in all of these patients, we had a steep decline in the total lymphocytes as a measure of the impact on CLL burden. So the venetoclax was working in all of these patients. And note, again, this is a log scale. So these are really a lot of cells are being killed by venetoclax as we move through that dose escalation. Now, we could identify some heterogeneity among CLL cells. I won't go into that for this study because we could identify major populations that had been previously published about the recirculation kinetics of CLL in the blood. But what I can tell you is that all of these populations were equally sensitive to venetoclax when we were able to fractionate them. So we used a range of approaches to resolve what are the key changes in the network underlying the response to venetoclax in CLL cells. And the most striking and consistent was the increase in the amount of BCL2 itself in the CLL cells that remain. So we're plotting them here. Is in gray is the profile of BCL2 in CLL cells taken from 3 different patients here. And in orange, we're overlaying the expression of BCL2 in CLL cells after that 200 milligram dose of venetoclax, just before they go on to the higher dose. And you can appreciate a big shift in the level of BCL2. And we've quantified that for all patients here, showing a fold change of BCL2 of MFI, median expression, at this dose. And you can see that by far the biggest increase we see is in BCL2, although there are some significant but modest changes in those related pro-survival proteins, MCL1 and BCL-XL. A consequence of this change is that the CLL cells we recover from patients after this dose of venetoclax are relatively less sensitive to venetoclax. So we're looking at a depth curve here of cells cultured in the presence of varying a titration of venetoclax, and you can see that compared to screening, this dark purple shows that the IC50 has greatly increased in CLL cells taken from this patient after venetoclax treatment. And for all the patients, we're summarizing the data on the bottom here, and you can see a modest but substantial increase in the IC50 of cells taken after they've been receiving venetoclax. So we wanted to know how these cells have developed a pro-survival high phenotype. Now, one of the hypotheses we had was, well, maybe venetoclax is best at killing those cells with lower BCL2, which you might predict. And we found evidence of that. We were also curious about whether there might be an extrinsic factor that drives up the levels of BCL2 in these cells. And one of the reasons for that is you'll note the distribution of these histograms here in orange. The upregulation we observed goes well beyond the range that we saw before venetoclax was present, suggesting to us that maybe there's something that's coming back and acting on these cells to drive the levels of BCL2 beyond what we saw before. So for that, we turned to mouse models. We needed an in vivo system where we're able to control the response to venetoclax and see if we could find evidence for there being an extrinsic factor. And it was known, for instance, that naive B cells from mice and humans are highly sensitive to venetoclax as well. So that was the cell population we went after. And so on the left here, I'll just show you the pathway, the impact of venetoclax on the pathway in BLAC6 mice and cells from BLAC6. And we predict that venetoclax targets BCL2 to relieve its inhibition of BAC and BAX, which could then go on and activate the apoptotic cascade. But we also made genetically modified mice that lack both BAX and BAC only in B cells, using a CD23 CRE to delete flox alleles of BAX on a BAC knockout background. These will be venetoclax resistant because although venetoclax can still bind to and inhibit BCL2, there's no BAC and BAX in the cells, so they'll never be able to activate that apoptotic cascade. And because we're interested in that dose escalation, that early phase, we subjected mice to just 7 days of venetoclax and then observed the impact on splenic B cells. That data is plotted here. And so cells from a BLAC6 mouse, you can see the B cells are highly sensitive. They at least 2 to 3-fold decrease in their numbers in the spleen and other organs. And you can see we could recapitulate this effect on BCL2 with this upregulation of BCL2, just like we observed in the cells from CLL patients. If we do the same experiment in these BAX/BAC deficient mice, there's no change in the number of cells and no increase in the level of BCL2, which you predict, right? Because these cells are resistant to venetoclax and unable to undergo cell death. And this confirms to us also that what we see with BCL2 increase is not simply a stabilization of the protein when it's engaged by BCL2 venetoclax. Okay, but we wanted to know though whether if there's an extrinsic factor, perhaps it might be linked to apoptosis. So we wanted to have a situation where there were cells resistant to venetoclax but in a scenario where there's apoptosis undergoing. And so for this, we made mixed bone marrow chimeras. So we took bone marrow from wild type and these BAX/BAC deficient cells, put them into lethally irradiated recipients, and waited for the B cell compartments to arise. And here we predicted that under venetoclax pressure, we will see apoptosis of wild type cells, but because they're resistant, we could read out the impact on these BAC/BAC deficient cells, whether there's a change in BCL2. And so here's some of the data showing that indeed the system worked. So in gray are the wild type cells in a mixed hematopoietic chimera receiving either vehicle control or venetoclax. And you can see that as we'd predict, there's a decrease due to apoptotic cell death in the wild type cells, but there's no change in the number of BAX/BAC deficient B cells in the same mouse. So what happens to BCL2? Well, now in the wild type cells, like we'd expect, levels of BCL2 substantially increased. And then interestingly, we see the same in the BAX/BAC knockout cells taken from the same chimera, in contrast to what we saw in the complete BAX/BAC deficient setting, where there was no apoptosis at all. Consistent with the notion that there is indeed some extrinsic factor that's elicited by the cell death of wild type cells in the environment that drives BCL2 in some manner upstream. So the answer to this question is yes, there's indeed extrinsic factors that drive this pro-survival high phenotype. Now, there's been many factors that are produced by the niche that affect CLL cells that could plausibly drive this increase. But the one we focused on immediately was BAFF receptor. This is a well-known regulator, a cytokine that regulates the homeostasis of naive B cells. And so if we first off ask whether BAFF is changed in these mice, we look at the wild type mice, you can see in this Eliza of serum that indeed the BAFF levels are increased in the wild type mice, but we don't see that increase in the complete BAX/BAC deficient mice receiving venetoclax, consistent with the fact that there's no apoptosis and no change in the levels of BCL2. So then what we did was go back to our mixed setting where we could treat mice containing a combination of wild type and BAX/BAC deficient cells with venetoclax. And here we're showing the control data where that drives up the level of BCL2 in the B cells we could recover. And then we took advantage of a mouse we made that it's BAX and BAC deficient, but also now lacks the BAFF receptor. So it's unable to receive that key growth factor signals. And you can see in this situation from the same experiment, unlike the controls, we don't see that upregulation of BCL2, indicating that indeed BAFF, which is increased in the serum of these animals, is driving this upregulation of BCL2 in the B cells that remain in these mice. And so that's just plotted here, but I'll skip through it in the interests of time. So of course, we're interested in knowing whether this happens in people. And so what we're able to do is take advantage of three distinct clinical cohorts. And the first I'll introduce here, these are breast cancer patients that for a clinical trial led by Jeff Lindeman here at WeHi, were receiving venetoclax for the treatment of breast cancer. We were able to take blood samples at various stages of screening, early phases post venetoclax or late phases when they've been on venetoclax for a long time, and study the impact on B cells, just like we did in the mice. And we don't have a confounding variable in there being a blood cancer present in these patients. And so consistent with what we'd expect, we see a decrease in the number of B cells in 4 patients that were taken after venetoclax, shortly after venetoclax treatment. And that's coincident with an increase in serum BAFF in all of these patients. And likewise, we see a modest but significant increase in the levels of BCL2 in the B cells after venetoclax treatment in these patients, consistent with the hypothesis that by driving B cells down, you get a coincident increase in BAFF levels that drives higher levels of BCL2 in the cells that remain. In the second cohort of patients, we took CLL patients that were receiving just venetoclax as part of their therapy. And so here, instead of a paired approach, we have pools of CLL patients at screening in blue, those patients receiving the venetoclax dose escalation, so the early phases of venetoclax treatment, and then a bunch of CLL patients that have been on venetoclax long-term to control their CLL, so beyond 2 years, shown in purple. What we're plotting here is the percentage of CLL that we could detect in the blood, and you'll appreciate that it decreased markedly by venetoclax in all patient groups. And then if we look at serum BAFF, again, we see this increase in the levels of BAFF that we could recover in these 4 patients compared to those that had yet to receive venetoclax. this is reduced or muted somewhat in those that have been receiving venetoclax for a very long time. But if we then look at the levels of BCL2 in the CLL cells that remain, the few CLL cells that remain, we can see that the levels are much higher in those that have received early after their venetoclax treatment. That corresponds with this increase in BAFF, just like we saw in the mice. So the scenario that's coming is that as we deplete B cells or CLL, there's reduced competition for BAFF or increased levels of BAFF that drives up BCL2 proteins in the cells that remain. So if that scenario is true, then you would predict that that would happen in any successful targeted therapy setting. Then maybe there's not something special about venetoclax in this context, that if you just reduce CLL in another way, you would expect to see increased BAFF and increased BCL2. So we're able to take advantage of a new combination therapy clinical trial in CLL patients where they receive an anti-CD20 antibody, obinutuzumab, as a lead into a combination of venetoclax and obinutuzumab. So that's shown here. The obinutuzumab only phase is shown in gray here, and you can appreciate that's very good at killing CLL cells as well. They markedly decrease. And then when you combine it with venetoclax in some patients, it improves the response even further. If we assay the levels of BAFF in the serum of these patients, we can see, coincident with this decrease, obinutuzumab could also drive up serum levels of BAFF in these patients. And in some of the patients, that would increase even further with venetoclax treatment. If we look at the levels of BCL2 now in those CLL cells that are remaining, we can see that obinutuzumab, again shown in gray, was able to drive up the levels of BCL2 in all of these patients to varying extents, but in some it was quite an impressive upregulation. And then the addition of venetoclax on top of that, which could drive CLL down even further, would also increase BCL2 levels in the remaining cells. So that's nice evidence, I suggest, that this scenario we imagine where venetoclax is killing cells or other targeted therapies are killing CLL cells very effectively, it drives up the bioavailability of BAFF, or in some manner might drive up the overall levels of BAFF, such that those cells that remain are able to access this key growth cytokine, and that drives up the expression of pro-survival proteins, in particular BCL2. And as I showed you earlier, we know that this reduces their sensitivity to venetoclax, and then might lead to this pool of CLL cells that are able to remain. And then over time, those accumulate further mutations that lead to therapeutic resistance and relapse. And so this is recently accepted, so it should be published soon. In another study, we showed that if we look at the normal immune cells, the T cells and NK cells from these same patients, we find that they retain their number very nicely. And likewise, their effector programs, the expression of immune checkpoints and everything remain intact. They're still functional, suggesting that further treatments that are able to co-target both pro-survival cytokines, just like BAFF that I showed you, and also immunotherapies are likely to be successful combinations with venetoclax and other targeted therapies to try and improve those early responses and prevent therapeutic relapse. And so with that, I'd like to just acknowledge the people that drove the work. So Kaylin Zhao was a key player in that thymus study I presented at the beginning, and Karis Tay and Mengjiao Luo led the CLL and venetoclax early adaptation story. We've got a bunch of wonderful collaborators that I'll just show here, and we're supported by a range of different funding. So I'd like to acknowledge them. And with that, I'd be very happy to take any questions you might have about any that I presented just now.